Morning everybody, welcome to Everyday Science. So I've been trying to stream on Twitch, but that hasn't worked out so well, so instead we're going to do a video. So we're going to be imaging proteins today. So I infected some tobacco plants the other day, and you can see the infection sites right here, these little spots on the leaves. And these guys are going to be expressing different fluorescent proteins. So we're going to talk about fluorescence, right? So these are proteins which glow when you hit them with a laser, and they can do different colors. So these proteins are going to be transiently expressed. And so what that means is that they're only going to be expressed or being produced by the cell for a couple days after I do the infection. So it's been about 2 days. And so what I'm going to do is I'm going to take little samples like this. So we're going to maybe switch you guys over and get you up close and personal to, let's see. All right. So, switch you guys around. Okay. So, hopefully you can see the bottom of the leaf nicely like that. And my lovely face, haha. So, we're going to take some little samples here. Like so. Going to cut those out. So, I'm going to be comparing my favorite protein and where it goes in the cell with a protein from a bacteria. And the bacteria I study is called agrobacterium, and it can insert new genetic material into the plant cell. And so that's how I got these different proteins to be expressed. But as I said, it's only temporary. So we got to image those today. Alright, so they'll switch you guys around. So now I've got my little samples right there. Let's see if we can get it back into focus. Alright, there we go. And so they're sitting like there. And so what we're going to do is we're going to make ourselves a little sandwich. So we can only use, on this microscope, a little bit of material at a time. So I'm going to add just a little bit of water. Okay, float those guys in. And so we're using a little microscope slide right now. And so I'm also going to take, see, if I was better at this, I would have gotten this ready ahead of time. I'm going to take a little cover slip, okay, a very thin piece of glass, and we're going to stick that right on top, like so. And then there's going to be a little bit of air bubbles in there, so we're going to smoosh it down. Okay, so we've got our little sandwich. Alright, and so afterwards we are going to need to seal the cover slip onto the slide to make sure that it doesn't fall off. So I'm actually going to use nail polish for that. Yeah, today we're going to use red. And so I'm just going to put that on the edges of the slide here. And put that on all 4 slides and seal it up. So I'm going to get the rest of my samples ready, and then we'll go to the microscope. Okay everybody, now we're over here at the Confocal Microscope. So we're talking about confocal. And so basically what that means is that we are able to see very, very small things with high resolution or very high detail. So over here is the lasers, right? Here's the lasers. And they're controlled up through here. And so what happens is the laser light comes back through here and goes through this box. And so there are filters which divide the different laser light. It comes into the microscope and it goes up through the objective. So these are the objectives right here. And so the objective is where the light comes up. So this is what's called an inverse microscope. Basically what that means is the objective is on the bottom and the light comes up. Normally other microscopes you see, they go the other direction. So that's one of the reasons we had to seal our sample earlier. And that's because what we're going to do is we're going to flip it over, right? So when we do the imaging, the laser light is going to come through here, and it is going to cause our fluorescent proteins to activate. So it's called excitation, right? They get excited and they go and they start glowing. And then we will pick up the emission frequency. So the emission is the wavelength in which that they are showing light. Okay? And so I'm going to be using a water objective, so I'm going to have to put a little bit of water here on top of the objective before I do my sample. So once I get the images and get that all set up, I'll show you how it works. Okay everybody, so we got some mood lighting. So you can see up here, that's the laser going through my sample. And I know it's dark, but that's just because we have to use the fluorescence imaging. And so here are my cells. So my favorite protein's this sort of yellow one here. It's forming this big glob. And this blue right here is the nucleus. So the nucleus in the cell is where all the genomic DNA, all the genetic material is. And so I'm going to add on top of that the transmitted light. And so we're going to add that there. And so let's see if this works. So, not quite seeing it, but that's okay. Ah, there we are. Oh no, it's too much. I'm gonna back that off. Doo-doo. Much less, much less. Okay, so when we layer like this, we can start to see the edges of the cell. And so these are where the edges are. So I'm looking very specifically at this particular spot. But since I'm only interested in the fluorescence part, we're just going to take that out. Okay, and there we go. So that's kind of how we do it. So I'm going to do a bunch of cells and I'm going to look and see if this is consistent and this is what we see all the time. And then, so we can look at these two different signals at the same time, and then we can layer them and then see where they are in relation to one another. So I hope that this was educational, and I hope that you'll enjoy other videos we do in the future. Follow me on @EverydayScience2. So instead of an E at the end of science, it's a 2 on Twitter. And message me if you have any questions. Thanks, and have a great day, everybody.